Good morning and good afternoon. On behalf of IUFRO and the coordinators of Working Parties 7.03.05 and 7.03.16, I would like to welcome you to this webinar in our webinar series on the behavioral and chemical ecology of bark and wood-boring insects. For those of you who are new to IUFRO, the International Union of Forest Research Organizations, this is a global, non-profit, non-governmental science organization. This global organization includes approximately 15,000 members from 120 countries and 600 member organizations. IUFRO is made up of several divisions, and the working parties responsible for this webinar series reside within Division 7, Forest Health. The coordinator of Division 7 is Ecki Brockerhoff, and within the division there are two research groups, Pathology, coordinated by Todd Ramsfield, and Entomology, coordinated by Marta Klepwick. These research groups are made up of working parties, and the working parties that host this webinar series, as mentioned, are 7.03.05, the Ecology and Management of Bark and Wood-Boring Insects, and 7.03.16, Behavioral and Chemical Ecology of Forest Insects. The mission of IUFRO is essentially the same as that of these working parties in this webinar series: to advance research excellence and knowledge sharing and foster science-based solutions to forest-related challenges. One of the many things the current pandemic has made clear to me is that our most meaningful resource is our networks. For those of you who would like to learn more about IUFRO and how to become involved in this powerful research network, I encourage you to consider one or more of the options listed here. Before we get started, a little housekeeping. Josephine Kefelec, a senior PhD student at the Forestry and Agricultural Biotechnology Institute at the University of Pretoria, will moderate this question period. For those of you who would like to ask a question, you can do it one of two ways. You can either use the raise hand feature in the chat box, in which case at the appropriate time, Josephine will unmute your microphone and allow you to ask your question directly of the speaker. Alternatively, you can type the question into the chat box and post it for everyone to see, and at the appropriate time, Josephine will ask your question for you. To make Josephine's job a little easier, I ask that you only post questions to everyone in the chat box, and if you have personal communication you'd like to do, please do that privately. I also remind you that the webinar is being recorded and will be posted after on the Working Party YouTube channel listed here. Before we get started, special thanks need to go out to a lot of people, most notably to Quentin Wignard, who is a senior PhD student also at the Forestry and Agricultural Biotechnology Institute at the University of Pretoria, who manages the IT side of the webinar series, and as mentioned previously, Josephine Kefelec, who will moderate the webinar series, and to the Forestry and Agricultural Biotechnology Institute at the University of Pretoria, or FABI, as many of you likely know it, for providing the platform from which we provide the webinar series. It gives me great pleasure to introduce today's symposium coordinator, Dr. Ecki Brockerhoff. Dr. Brockerhoff is well known to this community. He recently joined the Swiss Federal Institute for Forest, Snow, and Landscape Research, where he is the research unit leader for forest health and biotic interactions. He has had a highly productive and diverse research program for many years, covering the disciplines of entomology, ecology, biological invasions, and biodiversity. Dr. Brockerhoff is also very active within IUFRO, where he currently serves as coordinator of Division 7 Forest Health.
Hello and welcome to this webinar entitled Behavioral and Invasion Ecology of Hylurgus ligniperda. This is the introduction. My name is Ecki Brockerhoff. I'm with WSL, the Swiss Federal Institute for Forest, Snow and Landscape Research. I'm also an adjunct associate professor at the University of Canterbury, and I used to work for the New Zealand Forest Research Institute. I'd like to thank the organizers for the opportunity to contribute, and I'd also like to thank my many colleagues and former students who contributed to this research. Hylurgus ligniperda is a member of the Hylurgini. This is the tribe that includes the more prominent species in the genera Dendroctonus, Sintomicus, and others. It develops only under the bark of pines, very rarely other conifers, and it breeds in the lower stem, upper roots, stumps, and slash, especially when they're in ground contact. It feeds gregariously and has only visible galleries at low densities. And because it is a saprophytic species that colonizes only dead pine material as long as it's fresh and not too dry, it is not considered a pest in its native range. So it is native to Europe, the Mediterranean area, and also the Asian part of Russia. However, it is not well studied in its native range, and a search of Scopus reveals there are only 70 publications on Hylurgus ligniperda, and most of these not from its native range. This compares with more than 10 times as many papers on Ipstivographus, for example, and also many more publications on a number of other more prominent bark beetles. So what's special about Hylurgus ligniperda? It happens to be the most invasive true bark beetle, and it also is one of the most commonly intercepted bark beetles, or intercepted by border phytosanitary personnel. Like data from New Zealand, the US, and China show it is very, very often intercepted at the border. So I already mentioned the native range. This basically has a Palearctic distribution shown here in green on the map. However, there's some uncertainty about some parts of the eastern range here, like in eastern Russia, Siberia, northern China, and Japan, where it's not clear if it's native or present at all, or in fact introduced. However, it has first been reported as a non-native invasive species in Australia already in 1942, and subsequently it has been found in Uruguay, Brazil, South Africa, New Zealand, Chile, the state of New York, California, in Argentina, and apparently it's also present in Sri Lanka. So as a result, it pretty much has colonized most of the native and non-native range of pines, whereby the non-native range of pines essentially is in the southern hemisphere, where there are plantations of pines such as Pinus radiata. And here you can see an image of such a plantation, often very large expanses. And in New Zealand, there's about 1.5 million hectares of Pinus radiata, or Monterey pine, which is native to California. And this, in fact, represents a huge resource with very little competition from other insects. So as a result, Hylurgus ligniperda is often very abundant in its invaded range, such as in Uruguay, in Chile, and in Australia. And here are some examples from New Zealand, where 60% of all bark beetle catches in a nationwide trapping survey represented Hylurgus ligniperda. And it is about 7 times as abundant or more abundant than Hylastes aeder, which is the next most abundant bark beetle in New Zealand. And this example here, in 1 year study by Kerr et al., over 250,000 of Hylurgus ligniperda were trapped in just a single year. So this is a typical invader situation, where basically the availability of an underexploited, very large resource combined with the release from natural enemies allows this great abundance. And in relation to this, I would like to mention 2 points. First, that this creates some phytosanitary issues. And second, that this also represents tremendous research opportunities, of which I will show a few examples. So regarding phytosanitary issues, log and timber exports from countries like New Zealand are a very important economic activity. And here you can see the scale of this, where these huge pine log piles are shipped to other countries that import pine logs. And you can see the log stacks on the top right are covered in a sort of tarpaulin, And these are logs where fumigation with a fumigant takes place to basically disinfest these logs from any insects. And this has prompted a number of research studies to improve the processes of disinfestation and also to reduce the use of fumigants. However, I'd like to talk about a few other research opportunities, and in fact, Hylurgus ligniperda can be considered as a model invader. So an ideal model to study its biology and its invasion ecology. And in relation to this, I'd like to give a few examples as Hylurgus ligniperda can be a vector of blue stain fungi, such as these listed here. I'll talk a little bit about host finding, attractants, and trapping. And finally, an example of a study on temporal, spatial, and population patterns in relation to resource availability. Regarding blue stain fungi, my former master student, James McCarthy, did a study where he compared caged and uncaged logs and monitored the development of blue stain and the colonization of bark beetles. And he recorded these data for basically segments and sections of these pine logs, and results are presented here, where you can see blue stain on uncaged logs on the left, and this was much more abundant than on caged logs on the right. And you can see that the bark beetle colonization shown here in red in the lower graphs was clearly correlated with this sap-stain colonization pattern. We also know that bark beetles can be vectors or carriers of blue stain spores, and so obviously this is one aspect where, even though it's relatively harmless, Hylurgus ligniperda can have some impacts that are unwanted. Next, regarding attractants, basically the host volatiles alpha-pinene and ethanol are phenomenally attractive, and trap catches are approximately 200 times greater to these attractants compared with unbaited traps. And you can see that at the bottom in this graph. On the left, catches to unbaited traps, and on the right, catches to alpha-pinene ethanol, with using a range of traps. And also note the different scales on the y-axis on these two graphs. Basically, such attraction is comparable to a very good pheromone, and it basically means that these beetles are very good at finding their host resources, which probably contributes to their successful invasiveness. And also, trapping with these attractants is an excellent method for population monitoring. So trapping was also used to study the temporal and spatial population patterns. And basically in this graph here, you can see trapping that began in 2009 in old forest, and essentially no beetles were caught there. And as soon as the trees were felled, and this was a clear felled area, the catches increased phenomenally, and these lasted over up to 4 to 5 years. And it's a clear example of a resource pulse from harvesting and the availability of the slash that's following on, that this drives the population increase. This was work mainly done by my colleague Steph Sopo in New Zealand. So on to the other presentations in this webinar. So next up we have Nicolas Meurisse from the New Zealand Forest Research Institute. He will talk about dispersal of Hylurgus ligniperda. Then Kevin Chase, my former PhD student, who studied Hylurgus ligniperda in New Zealand and did some comparative studies in the US to study the role of propagal pressure in the colonization success of these species. And finally, Lea Bischofberger, a master student who is currently working with me here in Switzerland, will be presenting her results on the population genetic evidence for the invasion routes of Hylurgus ligniperda. So over to the next speaker then, and thank you again for your attention.
Hello, thanks everyone for attending this seminar today. My name is Nicolas Meurisse. I am working at Scion, the New Zealand Forest Research Institute. This talk is about the dispersal of the pine bark beetle Hylurgus ligniperda. Here, I am going to present some generalities on that beetle flight activity, and also some results from field trials that our team conducted in Pinus radiata plantation forests. Hylurgus ligniperda has colonized most pinus growing regions worldwide, including in New Zealand, where it was first found near Auckland in 1974. Now it is widespread in the entire country, and certainly one of the most commonly found insects in pine plantations. It is non-aggressive and saprophytic, most commonly found on dying pine trees or dead branches, fallen tree stems or stumps. Hylurgus ligniperda, however, remains a pest of quarantine status for log exports, notably as a potential vector of black stain root pathogens. This concern is illustrated here with the large amounts of logs stacked at the New Zealand port. Some of these logs may still have bark on them, and they may carry bark beetles, including Hylurgus. From systematically applying phytosanitary treatments to these logs, such as log fumigations, we are increasingly considering more targeted approaches based on the actual level of risk. And, in this context, some fundamental knowledge of insect ecology and biology is much needed. Hylurgus ligniperda, because it is widespread, is a key base of concern, and we aim to better understand the probability of colonization of pine logs by this species. This can happen in the forests and along the export chain as the logs transit to the ports. Different factors influence the level of activity of Hylurgus ligniperda, such as its local abundance, its seasonality, and its dispersal capability. Here are results from an extensive trapping network set up between 2013 and 2016. This work was led by Steve Posen and conducted by our entomology team at Scion. Hundreds of flight traps, baited with ethanol and alpha-pinene, have been set across the 8 main forest regions of New Zealand. They were regularly checked during 3 years and a half. This was a huge effort, totaling more than 17,000 days of active trapping. Now when you look at the graphs on the right, these show you one of the main learning from that network: the probability of Hylurgus flight depending on the time of the year. And there were significant regional differences. In the warmer regions in the north, the level of risk is high the entire year, while in the colder southern regions there is a strong seasonality. From the picture of the trap, you will also notice these have not been set up in the standing forests. Instead, these were placed in recently harvested stands, as these locations contain the largest amount of woody debris colonized by bark beetles, and from which they emerge. In New Zealand and elsewhere, we have already learned quite a bit on the flight activity of Hylurgus ligniperda. From the previous slide, we have seen Hylurgus is seasonally active in colder areas, and in most regions we can infer the following pattern: a peak of flight activity in the spring, fewer flights in summer, and then another increase in activity in autumn. The time of the day also influences activity, most flights occurring a couple of hours after sunrise, but they continue all day. And then, as you can expect, there is a strong influence of meteorological conditions. Temperatures above 12 degrees are associated with higher probability of flight, and high wind, rain, or humid conditions are detrimental. You can see some variables in the diagram on the right. These are sorted in order of decreasing overall influence on flight activity, as estimated from a recent modeling work based on Bayesian network. Now, moving back to our plantation forests, This is how a typical plantation of Pinus radiata looks like in the central North Island of New Zealand, with a mosaic of stands of different ages and recent clear fells that contain exceptionally large amounts of woody debris colonized by bark beetles. To give you an idea, it is estimated that a typical clear fell contains about 350 cubic meters of deadwood per hectare. In comparison, a 25-year-old stand produces only about 10 cubic meters per hectare and per year. Recently harvested stands thus represent significant source locations for bark beetles, such as Hylurgus, and, as you could see from this other graph on the right, the highest abundance is in the first year following harvest. This is the green bar. After that, it progressively declines as all the dead branches and stumps degrade. After 4 years, the clear-felled area doesn't act as a source location for bark beetles anymore. There are also sink locations which attract these beetles. In standing forests, these can be the occasional dead or dying trees or fallen branches. But in the plantations, other attractive material are the pine stems, as they get felled and remain on skid sites for a couple of days or sometimes weeks. And then, in the clear fells, large amounts of leftover branches and stumps also provide a particularly attractive habitat to bark beetles. What is largely unknown, however, is the dynamic of movements of beetles between these sources and sinks. We can observe Hylurgus adults emerge in large quantities from old stumps, but we don't know how far they go. And, on the other hand, we can occasionally see large numbers of Hylurgus landing on the cut stems, but we don't know where they come from. Trapping experiments can be informative about dispersal. For instance, as we place transects of traps at increasing distances from a source location, we expect to see a declining pattern of captures as beetles can only disperse up to a certain distance, and also because of dilution effects. This is illustrated here from one of Ecki Brockerhoff's trial. Traps placed in a stand harvested 1 or 2 years ago are expected to capture relatively large amounts of beetles. And traps located in a stand harvested later, for instance 3 or 4 years ago, would capture significantly less as the source gets depleted. Here are the results. If you look at the graph on the left, this conforms to expectation. There is at least 3 times more captures in the stand harvested 2 years ago, the left circle, as opposed to 3 years ago, the right circle. And more strikingly, on the graph on the right, there are way more captures in the stand harvested 3 years ago compared to 4 years ago. That last one seems almost completely depleted of beetles. Also, if you look at intermediate record points, these ones around the 0 values on the x-axis, and that represent the edge between the two stands, you can see there are intermediate levels of capture, indicative of beetles' movement between the two stands. Now, aiming to more precisely quantify the dispersal of an insect is a bit of a challenge. Tools have been developed to track the movements of individual insects, such as flight tracking devices to use with flight gauges or harmonic radar for field studies, that is illustrated with the bee here. But these remain challenging, generally expensive and difficult to apply to track many individuals. One alternative is to estimate the spatial redistribution of a population. This can be done from a non-population source. For instance, with only one clearly identified source of emerging bark beetles in your entire landscape, and capturing individuals from that population at different distances. Or, in a more controlled manner, from the release and subsequent recapture of marked individuals. This is the method illustrated on the right here. A large number of marked individuals are released at a single point location, and this release location is surrounded by a large number of recapture locations, let's say traps with semi-chemicals in a bark beetle context. Then, the distribution of dispersal distances can be inferred from the capture data. We have chosen this later approach to quantify the dispersal of Hylurgus ligniperda. Mark recapture assays have been performed for a range of bark beetle species, but to our knowledge, never for a saprophytic bark beetle. We have done this in 2014 in a large Pinus radiata clay fell in the central North Island of New Zealand. There, we could find an ideal fit location for such a large-scale experiment. And here, an illustration of the trap setup, organized in concentric circles around a single release location. The traps were located up to about 1 kilometer. Then, there were 15 releases across the summer and autumn months, for a total of 18,000 beetles, of which about 9,000 flew. And of these, we caught only about 100, which is about 1%. This seems very low, but it is generally on par with other examples from around the world. Here, you can also see the marked beetles using a fluorescent pigment. There were different colors as we had two groups of beetles, flight experienced ones that we got from traps and flight naive ones that we just emerged from rearings. The picture on the bottom right is the release platform, and here is a little example where you can see these Hylurgus adults taking off. The results from these trials are presented here. The red dots on the map represent the capture locations on the grid, and on the main figure, for flight experienced beetles, you see the number of recaptured beetles per trap location declining with distance. Many traps did not capture a single beetle, but given the large number of traps, we got enough data to exemplify that typical declining with distance capture pattern. For the naive beetles, we couldn't observe that similar declining with distance pattern. In fact, we had comparatively low level of captures in the traps located nearby the release location. A result that in a sense was disappointing, but also informative, suggesting that emerging Hylurgus may require a period of flight to initiate chemotropic orientation behavior. Our second objective was to quantify the proportion of Hylurgus dispersing as a function of distance from the release location. What the graphics is showing is a parametric function describing the decline in density of the flying beetles with distance. Using parameters from this function, we could then construct the final dispersal distance curve, which is presented in the main figure. Less than half of the beetles can disperse 1 kilometer or more, and only around 5% of them, as predicted by the model, are able to disperse up to 3.7 km or more. Remember now that the trial itself was only at distances up to 1 km, so we must remain cautious with predictions for largest distances. This new knowledge of the dispersal potential of Hylurgus can be used to estimate the proportion of a source population that disperses from one location to another within the landscape. For species of economic or environmental significance, this can prove very useful in management initiatives. However, because of the dilution effect, each distant source population will usually contribute only few individuals to the flow of beetles arriving at a particular location. This is illustrated here, looking at the graphics on the right, where there is a source location in the middle. That is the one with a large beetle represented. There is a prediction that 16% of the emerging beetles will not move very far, less than 50 meters away. But then, if you look at a more distant location, let's say a 1 hectare area located 1 kilometer away, which is the tiny beetle in this example, the projection is that only 0.06% of the emerging population would settle there, showing the importance of the strong dilution effect. Consider now all these locations located at that 1 kilometer distance, which is the yellow annulus. There, it is estimated 4% of the emerging individuals would settle. Thank you for listening. This is now the end of this presentation. And this is just the last slide to thank all the contributors to our release recapture trials. And let's chat later if you have any question.
Hi there, my name is Dr. Kevin Chase. I am currently a research entomologist with the Bartlett Tree Research Lab in Charlotte, North Carolina, United States. Today I will be discussing Allee effects and invasions of Hylurgus ligniperda and the role of tropical pressure. I want to thank Ecki Brockerhoff for the invitation to do this presentation. Before I begin, I first have to thank my co-authors, Dave Kelly, Ecki Brockerhoff, and Sandy Leopold. I also have to thank the 3 fantastic technicians that helped me complete this project, Nico Mandel, my brother Brian Chase, and Martin Bay. This project was funded by the Better Border Biosecurity and also the Brian Mason Scientific and Technical Trust. I am very grateful for their support. Most invasive species stories have an invasion pathway component. Many tree-infesting invasive species stories begin like this. A container ship is loaded up with a variety of resources. Many of these resources either contain wooden products themselves or sit on solid wood packaging materials such as pallets. Inside of these materials may lie hidden a non-native tree-infesting insect, such as the beautiful Asian longhorn beetle. If a non-native species successfully establishes in a region with co-evolutionarily naive host species, the invader may have potentially severe consequences on the range of hosts they attack. This graph clearly demonstrates that the number of bark beetles invading the United States has increased sharply over recent history, largely tied to the drastic increase of global traded goods. But it is really important to note here that most species go undetected, and also that most species intercepted at ports of entry never become established, which leads us to our big question. What makes some bark beetles more successful invaders than others? Today we will discuss the bark beetles Hylurgus ligniperda and Ips pini, and what makes one of these bark beetles so successful and potentially why the other is less successful. This is a general framework for predicting the probability of establishment based on the 3 main bark beetle guilds. Of course, there are some species that can fit different guilds at different stages of their population cycles, and I'm speaking in very general terms. Hylurgus ligniperda is a true saprophytic insect that only attacks dead wood and has no pheromones that we know of. Therefore, this species is dependent on following only host volatile cues. This may be an advantage when arriving in a novel environment, as opposed to primary insects who may be searching for pheromones produced by mates that are not present in large numbers. We will be discussing Hylurgus ligniperda and Ips pini and their invasion success under the context of tropical pressure and the Allee effect. Tropical pressure simply refers to the total number of individuals that exist or arrive at a single point of place and time. The Allee effect is also called positive density dependence. If an Allee effect exists within a population, there is a threshold at which a species' population growth will be positive or negative above or below that point, respectively. Once below the Allee threshold, that population will trend towards extinction unless further recruitment or immigration occurs. The objective of this project was to measure what founding population size results in colonization success of Hylurgus ligniperda and search if an Allee threshold exists. I will also compare the study with a similar study we conducted for Ips pini. The Hylurgus ligniperda study took place on the South Island of New Zealand. The photo you are seeing is of the beautiful Avoca River Valley, which highlights one of the river valleys this study took place in. Pictures like this really make me miss New Zealand. The first step in the study was to find areas on the South Island without an existing population of Hylurgus ligniperda. To do this, we hung panel traps in remote river valleys situated as far away from any pine trees we could find. We hung panel traps paired with ethanol and alpha-pinene lures, host volatiles, to monitor for the presence of Hylurgus ligniperda. To our surprise, it was actually quite difficult to find areas without this insect. While there was a negative trend observed of beetle capture rate away from pine plantations and pine windbreaks, we still captured relatively high numbers of Hylurgus ligniperda at distances up to 50 kilometers away from the nearest pine tree, and in these remote river valleys surrounded by mountains. We collected enough data to publish these results in the journal Ecological Entomology. Fortunately, we did find 6 valleys that we either captured 0 or few enough beetles to feel comfortable they would have very little interference during our experimental releases, which is the next step of the project. With those sites established, we were able to move on to the really fun part of this experiment, conducting releases of different densities of individuals to search for an establishment threshold of Hylurgus ligniperda. We released wild-caught individuals and at first tested if we could use Dagaloo powder to ensure the beetles we recaptured were indeed the ones we released. However, the powder influenced the behavior of the beetles and we decided not to use it. We used 0.3 meter Pinus radiata logs and defined successful establishment as the presence of eggs or larvae found within the logs. Here is a visual image of the experimental design and densities of Hylurgus ligniperda we released. We replaced logs either 1 meter or 10 meters away from a central release point in the field. We also conducted 1 meter releases in a caged environment to remove the dispersal loss component of an invasion event. This forced beetles to a confined area where they can only interact with the source material and individual conspecifics present. Here is a time-lapse video of a release event with logs placed 1 meter from the release point. One thing you may notice is that it was a windy day. If you watch the beetle's crawling direction, it is to the left side of the screen, which is the direction the wind was coming from. When we collected these logs 1 week later, we observed that the log placed in the same direction the wind was coming from was heavily infested. This makes sense when thinking about the volatiles being emitted from this log in the direction of the wind, which would pull the beetles towards this log. On days without any wind, we observed that the beetles were more likely to immediately fly, as opposed to a crawling behavior like that observed in the video. After logs were collected, stored for enough time to let beetles mate, lay eggs, and larvae to be present, we dissected all the logs and recorded the presence or absence of the different life stages of F1 Hylurgus ligniperda. In the following 3 slides, I show the results from this study. I will show 2 graphs in each slide. The first graph displays the establishment probability on the y-axis, which is based on the number of propagals released, which is displayed on the x-axis. The triangle in the curve represents the inflection point of the curve that also represents the Allee threshold. Above this point, we expect growth to be positive, and below this point, we expect growth to be negative over time. The second graph displays the number of progeny produced per release density on the y-axis, correlated with the release density on the x-axis. A positive slope informs us that competition is not a strong force at these densities, where a negative slope would inform us that competition may have a negative impact on the population at greater densities. In the caged experiment, we see that Hylurgus ligniperda established at the lowest density released of 5 beetles, and that likelihood of establishment was strongly positive above this point. We also see that a positive slope exists in the per capita reproduction graph, indicating that no negative consequence on reproduction occurs at any level of release we conducted. In other words, they appear to be quite cooperative at these densities. In the 1 meter field release, we observed some colonization events at a release of 10 beetles. Colonization was observed 100% of the time at beetle densities of 100 or greater. Like the previous graph, we observed a positive per capita reproduction trend. In the 10 meter field release, we observed much fewer establishment events even at the highest level of beetles released, which was 500 beetles. Although the distance was only 9 meters more than the previous release slide, it appears that even a slight increase away from host material decreases the likelihood of establishment. We wonder if the crawling behavior versus the flight behavior may influence the success of these events. However, we also know from our previous study that these beetles are very capable of dispersing long distances and finding host volatiles, at least when placed in a trap. Because so few logs were colonized, the per capita reproduction graph is not very informative. We conducted a similar experiment for the secondary bark beetle Ips pini, which is native to the United States but has been intercepted, yet never established, across various global ports of entry. This beetle attacks stressed trees and utilizes a sex pheromone to find mates. Like the Hylurgus ligniperda portion of the study, we first needed to find areas without any Ips pini. We set up a transect of pheromone-baited funnel traps stretching from Logan, Ohio to Oskaloosa, Iowa, and we assumed that the corn and soybean fields of the Midwestern states of North America would contain no beetles. However, we were surprised once again how many beetles we found at locations very far away from any pine trees. Fortunately, we were able to find a spot that we captured 0 beetles at the very end of our transect. We used the same experimental release design that we used for Hylurgus ligniperda, with the main difference being that we did not include a 1 meter field release due to limitation of carrying enough Pinus strobi logs from West Virginia to Iowa every other week. We also increased the total number of beetles released to 1,000 because we were able to catch Its pini in greater numbers in the field. Another factor that is important to remember when viewing the results is that we counted adult beetles reared in cardboard tubes as opposed to dissecting logs and counting larval densities. We realize this is not a true comparison, and there are many logistical reasonings that account for these differences. In the cage portion of this study, colonization occurred at the lowest number of Its pini released of 2 beetles. Establishment probability, as measured by the presence of F1 adults, is also expected to be positive above 7 beetles. However, one striking trend that we observed was reduced per capita growth at higher numbers. If we only consider colonized logs, and not all the logs that are displayed in this graph, this trend is quite strong. The fewest beetles to establish in the 10-meter field release was 60 beetles, but we did not consistently see establishment occur until the 200 beetle release density. Like Hylurgus ligniperda, very few logs were colonized at this distance, and the per capita reproduction graph is not very informative. To summarize, we captured both Hylurgus ligniperda and Its pini at great distances away from host resources in their invaded and native ranges respectively. We observed successful establishment events of Hylurgus ligniperda, measured by the presence of larvae at least once at the lowest levels in all release scenarios. Additionally, we did not observe any negative consequence of per capita growth rate of larvae at high densities. It is likely, however, that we would have observed fewer adults than larvae if left until completion due to possible resource exhaustion. While we observed establishment measured as reared adults of Its pini at the lowest release density in the caged environment, a successful establishment event did not occur until 60 individuals, and high consistent establishment wasn't observed until 200 individuals. Native density dependence also may be a limiting factor for invading population of Its pini. We conclude that Hylurgus ligniperda is a successful invader based on its ability to find hosts and their ability to cooperate and reproduce large numbers of brood, even at high densities of adults. Its pini colonized field logs at higher densities and had a strong negative density dependent growth rate, which may be 2 reasons why this beetle has been a less successful invader than Hylurgus ligniperda.
Welcome to my master thesis presentation with the topic Population Genetic Evidence for Worldwide Invasion Routes of Hylurgus ligniperda. In my presentation, the superscripted numbers are indicating references. Hylurgus ligniperda is native to Europe and is getting distributed by trade in woodpacking material and timber. The first non-native population was reported in Australia in 1942. Other non-native populations are in Uruguay, South Africa, New Zealand, Chile, USA, and Argentina. The aim of my thesis was to get a better understanding of possible invasion routes and also to examine bridgehead effects with genetic methods. A bridgehead population is a non-native population that is spreading further into new non-native ranges. Literature indicates such a bridgehead effect for Australia, Chile, and New Zealand. For my thesis, I got samples from the native range in Europe, from France, Portugal, Italy, Hungary, and Greece. From the non-native range, I got samples from the US in California, Chile, Uruguay, Argentina, South Africa, and New Zealand. From these samples, I extracted DNA from the legs of the beetles to minimize cross-contamination with other organisms and parasites. To get more robust results, I used the nuclear and mitochondrial marker. For the nuclear marker, I used the RSTIN2, and for the mitochondrial marker, I used COI. In this presentation, I will show you the results from my haplotype analysis with the network and distribution, and also the discriminant analysis of principal components. The haplotype analysis for the nuclear marker RSTIN2 gave 110 haplotypes for the 233 samples. In this haplotype network, only the haplotypes including more than one sequence are shown. In total, there are 36 haplotypes, including 176 samples. From a primary introduction, we would expect shared haplotypes between the native and non-native population, as we see it here, and here. But there are also shared haplotypes between non-native populations, such as Argentina and Chile, Argentina and New Zealand and US, New Zealand and Uruguay. The South African population had no shared haplotypes with any other sampled population. The haplotype analysis for the mitochondrial marker COI gave 33 haplotypes for the 257 samples. In this haplotype network we see some similar patterns as we saw before with the nuclear marker aristin-2, such as shared haplotypes between Argentina and Chile, and also shared haplotypes with New Zealand and the US. Here it's also including the native population in Italy. But we see also some differences, such as shared haplotypes between Argentina and Uruguay, and the shared haplotype between France, South Africa and Uruguay. The European population is quite separated from the rest of the haplotypes. The haplotype distribution for the mitochondrial marker COI shows a high haplotype diversity in the native range of Europe. We would expect there a high haplotype diversity due to evolution of the species over time. In New Zealand we have 2 haplotypes which origin from the European population. The more dominant haplotype in New Zealand is the only one that occurs in the Californian population. This indicates a possible bridgehead from New Zealand to California. The Chilean and Argentinian population have no short haplotype with Europe. This is an indication for an underrepresentation of the European haplotype diversity. Chile and Argentina have several haplotypes in common. The discriminant analysis of principal components shows some similar patterns as we saw before with the haplotype analysis. For both nuclear and mitochondrial marker, there are overlapping clusters for population, such as the New Zealand and North American population, or the Chilean and Argentinian population, as we saw before with the shared haplotypes. Uruguay is separate from the other populations. For the nuclear marker, the European population is overlapping with the non-native populations, in compared to the mitochondrial marker, where the European population is differentiated and separated from the non-native population. In this map, the most likely invasion routes based on my analysis are shown. The question marks symbolize populations that are not included in my analysis, namely New York, Sri Lanka, China, Japan, and Australia. The arrows from the European native populations to the non-native range shows the primary introduction. Both the haplotype analysis and the discriminant analysis of principal components show similarities between the New Zealand and North American population and between Chile and Argentina. Since the most dominant haplotype from New Zealand is the only one that occurs in California, it's most likely that the New Zealand population is a bridgehead for the establishment in California. Also, the Chilean population seemed to be a bridgehead for the population in Argentina, since the Chilean population is much older than the Argentinian one. Literature indicates possible bridgehead populations in Australia, New Zealand, and Chile. With my analysis, I found indication for a bridgehead effect for 2 out of these 3 populations. Potential bridgehead effects occur from Chile to Argentina and also from New Zealand to California. However, my genetic analysis provides no information on the direction of population movement. Therefore, interception data and literature have to be included. as expected, shows the non-native population origins from the native European populations. My study is limited by the sampling size, in particular due to underrepresentation of the European population. Also, there are some interesting non-native population missing, such as the potential pre-child population in Australia and the Neurochron population in the US. The nuclear marker Aristin-2 provided me with quite some challenges due to a lot of UPAC codes at a few variable sites, which created a high variability of the sequences. However, the general patterns based on the COI could be confirmed with the nuclear marker. To complete the study and fill the missing gaps, we would appreciate to receive some samples of Hylurgus ligniperda, which are freshly collected or trapped and stored in ethanol. Please send them to Ecki. At this point, I would like to thank my supervisors and the collectors around the world who provided me with the samples to make this study possible. Thank you very much for your attention, and now I would like to answer your questions.
Thank you very much to all the speakers for your beautiful presentations. It was really, really interesting. So I think we can start the discussion period. So just a reminder that if you have questions for our speakers, you can post them in the chat box and I will read them for you. But please indicate which speaker the question is for, so I can ask the proper person. Okay, so let's start with a question for Nicolas. Was activity only measured through trapping, or have lab assays been also done? Ecophysiology for CT min, for example. Can you hear me?
So maybe Josephine, ask someone a question while Nicola works on.
Okay, it's fine. Yeah, Nicolas, we can't hear you, unfortunately.
We can't hear you, Nicola.
Yeah, I'm just going to ask another question in the meantime. So for Kevin, are both Hylurgus males and females equally attracted by alpha-pinene and ethanol, or is there a pioneer sex? How did you control sex ratios in the release and the cage experiments?
Yeah, that's a good question. As far as attraction, I'm not sure. After you catch them in a trap and they're dead, that's very hard. But the fortunate thing about these insects is that the males do actually make a call. They make a squeak. So that's very nice. So in the caged experiments, at the very low numbers, any number below 10, we actually did equal numbers, those sex ratios. But above that number, we just assume that there's a fairly equal ratio. There is a small bias, but as far as a pioneer sex, I am less certain about that. Ecki, do you know of a pioneer sex? I don't know for these species.
Well, apparently the females start the galleries, but I'm not totally sure about that. But
yeah.
I believe the sex ratio is pretty much 50-50. And I believe that's also more or less the case in trapped beetles.
Yeah. And Josephine, may I take the next question as well with the SIB mating? Is that okay?
Yes, go ahead, yes.
Yeah, so for SIB mating, we actually had started an experiment that unfortunately we had a power outage and all of the insects died while sitting in a fridge during this experiment. But what I can say about it from observation and from the start of that experiment is I do believe that SIB mating does occur, that as the beetles are developing and prior to leaving the log, that their mating does occur and SIB mating does exist. Based on the initial results that we got. Now, where when the insects died, we had observed larvae from siblings, directly from siblings, but as far as how viable they were as far as reaching adulthood and then further mating, we were not able to complete that part of the project. But as far as do larvae come from siblings that have mated before leaving the log, the answer I can tell you is yes.
So actually, to shed more light on that question, and so I believe the literature is kind of unclear on that, and I've wondered that for a long time too. But actually, there's I believe a PhD student, Pauline Daquin, who's with Jean-Claude Grégoire, and I believe she's a participant in the webinar. So perhaps if that's possible, if she would like to say something about that, we'd all love to know if you have any more insights. I think I did see her earlier, Pauline. Josephine, are you able
Yes.
to allow her to turn on her video and say something? In case she's, or alternatively, Jean-Claude.
Josephine, she sent the message.
Ah, yes, I see now. Okay, so she says, as Ecki Brockerhoff said, I am working on pre-emergence mating in Exogamous colitinae. My work carries on different species and implies spermatical dissections, rearing, and genetics. Faber and Carl have already reported pre-emerging mating in Hylurgus ligniperda, and I am going this spring to the southeast of France to analyze further pre-emergence mating in the species. This is an aspect of reproductive behavior that has already been highlighted in different exogamous species, such as Ips grandicolis, Dendroconus valens, and Tomicus piniperda, and I am sure that it is present in many more species.
Mm-hmm. Yeah, so thanks very much for sharing that. We are aware of that publication by Carl, and also Nicolas had mentioned he has observed that, but then others said that they believe that not to be the case. So I think some experimental evidence would actually be interesting. I don't know if Massimo Faccoli is present in the talk, but he said based on his expectation, he would believe that these young adults, before they emerge from the logs, from the galleries, and that they need to disperse first and do some maturation feeding. So yeah, I think that's not entirely clear yet, whether they in fact do mate and produce viable offspring if they do mate before they disperse. It would explain, however, if they were, why they are such successful invaders, because it would greatly help with colonization in an invaded range.
Thank you, Ecki. Nicolas, would you like to try your mic again? Yeah, we can't hear you, unfortunately. I'm sorry. Okay, so next question is for everyone. Is there any chance that Hylurgus ligniperda might affect weakened forest regeneration through its feeding as Elastis ater or Ilobius aviatis do?
So perhaps I can answer that, because we've also wondered about that. And in New Zealand, we have observed that Hylastes ater does in fact do maturation feeding on seedlings. And we've also studied that, and there's a paper by Sopo et al. That reports the results. And we've also seen that in most cases in New Zealand, Hylurgus ligniperda is more abundant in these kinds of situations because the seedlings are planted into clear-felled areas where there are very high populations of both of these species, but normally more Hylurgus than Hylastes. And all the seedlings that we've studied, there were quite a few that had signs of maturation feeding. And in every case, it was Hylastes, and we didn't find any Hylurgus in these cases. So I'm also aware of studies from South America that report that Hylurgus also does this, but we have not seen this in New Zealand. So I don't know if there is a case of misidentification potentially, or whether in fact the behavior is different. So, but in New Zealand at least, I have not seen that.
Thank you. I see Mike Wingfield has his hand up. Would you like to say something, Mike?
Yeah, thanks, Josephine. I'm going to stick on my video and hope it doesn't mess up things. Yeah, just it's changing this topic a little bit, but talking about things being different in different parts of the world. So Ecki, and most of my experience with this insect has to do with the fungi that it carries. And looking at, for instance, infestations in Chile, I saw. Can you hear me?
Yes.
I saw more or less what you're saying, sort of evidence of maturation feeding. So Hylurgus eta is there as well. But something that came to mind just listening to the talks, and thanks for them, they were all really interesting, is how the presence of other beetles might affect the behavior of, say, for instance, Hylurgus. So in Chile, Hylurgus seems to have replaced what, for instance, in South Africa, or in New Zealand, say, Ips grandiculus is doing for us Orthotomicus erosus. So what we would see as Hylurgus going into the basal parts of trees, not doing terribly much, but the space that I would see it infesting or invading in Chile is what Orthotomicus erosus would do here. So almost, I don't know what happened to Ips grandiculus in Chile. It was there originally, but it seems to have just disappeared, maybe replaced by Hylurgus ligniperda. And I think there are some people in this seminar that have looked at that situation in Chile. But it does, I mean, I do wonder whether the fungi associated with these insects might do something, but also, where the competition is changing the behavior patterns. Certainly, Chile to me look quite different to what I see here and what I've seen in New Zealand also, for that matter.
So maybe I'll just make a point regarding New Zealand and this potential for competition. So firstly, in New Zealand, there's no Ips grandiculus. That's only in Australia. It's not established in New Zealand. And in New Zealand, the only two bark beetles that are abundant enough to play any role in competition potentially are Hylurgus eta, which has been there for some 45 years longer, at least, than Hylurgus ligniperda, and was initially probably more abundant than it is now. And once Hylurgus ligniperda arrived, it sort of partly displaced Hylurgus eta. And we've also seen in bark sandwich experiments and observations that when you have a gallery of Hylurgus eta and you add some Hylurgus ligniperda to the same sandwich, then Hylurgus basically munches through the galleries of Hylurgus eta. And I am pretty sure that Hylurgus is the superior species in these cases where both species occur. So I believe that competition is not an issue. And I think they also both move around the same kinds of ophiostomoid fungi, which I think at least probably don't have any sort of essential role in the beetles feeding. So I could be wrong, but we've seen no clear evidence that they actually require the presence of these fungi. I think it's more that the fungi, blue stain fungi, catch a ride on them and get moved around like that. But that's something that would be worthwhile investigating further, certainly. But maybe someone can say something about the situation in Chile or Uruguay, for that matter.
So Ecki, I was thinking more not competition between Hylurgus and Hylurgus, but if, for instance, Orthotomicus erosus, which seems to be doing what Hylurgus more or less is doing, what I've seen in some situations in Chile. But I mean, this is just sort of anecdotal.
Yeah, so I can't say much about Chile, although I'd love to visit. But I don't know, who's there from Chile who would like to share something on that? Or anyone else, like maybe Jerry Hulzer has some insights, because I believe he has some people in his lab who are also interested in this species.
Hello. We have not really worked on this beetle very much. We just isolated some fungi and such, but its ecological significance, especially in Chile, we have no idea. I'm just amazed by what you are all saying about its ability to displace or suppress other species occurring in the same microhabitat. It's pretty interesting. It reminds me of the story of the Cyrex wood wasp that seems to be suppressed after the arrival of almost any bark beetles that carry Ophiostomateles. So perhaps we are seeing sort of the knock-on effect of arrival of some fungi that are able to suppress other bark beetles that require fairly fresh wood, and perhaps Hylurgus just serves as a vector. It's not really the effect of the beetle itself, but it serves as a vector. It's this sort of a pig of a bark beetle anyway. It really lives in this horrible wood sometimes. And perhaps it also vectors fungi there, or maybe many fungi, maybe just the sheer diversity of Ophiostomateles and other fungi that are not really good to bark beetles. Maybe just the fact that it vectors a lot of them has this effect of knocking down other wood borer populations. Perhaps worth looking into some more.
Thank you very much. Ecki,
Can you hear me now, Josephine?
Oh,
Yes,
yes, great,
Nicolas.
fantastic.
Okay,
Okay.
You may need to
so-
repeat the question.
Yes, I will
You
repeat the
couldn't
question
remember?
for the audience anyway.
I don't.
So the question was activity only measured through trapping, or have lab assays been also done? Ecophysiology for CT-min, for example.
No, we mostly did trapping or flight activity. Yeah.
Okay, so I have another question for you. The release capture assay has an issue. The farther you go, trap density drops. Can you comment on that?
Yes, that's very difficult to trap at increasing distances, and that's one of the reasons we could only set up traps up to about 1 kilometers, and even at that distance, we had very few recapture. I think we only had 1 or 2, and the only solutions to that are to increase the number of traps at these distances or release very large amounts of beetles. But in either way, it's very difficult, and that is a known limitation of mark recapture studies. Yes.
But maybe if I can add something to that.
Yeah.
You did correct for that mathematically, right? Like you correct for the
Yes,
area.
the mathematical interpretation, correct.
The trap density that decreased over increasing distance from the release point, you corrected for that in the analysis, right?
But you still have lots of zeros. And if you were to trap for distance, let's say 5 kilometers, you would probably just end up with having no captures, even with many traps, large amounts of beetles and effective traps.
You just need to release more and have even more traps.
Or set them in remote valleys far away from any pine trees.
Yeah, where there's less competition. Because Kevin,
that's another problem. Yeah.
Even a long way away from any pine trees.
Yes. In one observation, I had traps in the podocarp forests of New Zealand with no pines anywhere nearby. The nearest pine plantations were 45 kilometers away with 3 mountain ranges. And in the midst of that podocarp forest, you put up a trap and they would still find them. It was just unbelievable.
That's good. Yeah.
They are very effective at finding their source if there's nothing around.
Thank you. I had then a question for Kevin. How do you think interspecific competition might affect the establishment probability of Hylurgus?
Yeah, that's a good question. That's another aspect I wish we could have continued on. As Yuri just mentioned, they're kind of pigs of insects, and they're happy to go after the scraps. So I think part of the reason that they're so successful is they'll utilize stuff that, from a saprophytic standpoint, that a lot of other beetles can't. Whether it's a moisture content standpoint or something else, I don't really know. But you really got me thinking, Mike Wingfield, about Ipsgranicolis. I think Australia would be the perfect place to do that with Ipsgranicolis present there and Hylurgus ligniperda, as well as maybe up in New York where there's a population. I've been working with Ipsgranicolis recently, or grandiculus, as some people say, and I would love to see that. I think, I guess I don't know. I would just love to see the data on that. I don't want to speculate beyond that from a direct competition standpoint. But they are very effective at utilizing degrading material. Ecki, you got anything else there, or Nicola, or Lea, or anybody else?
No, I mean, we've done a few studies, and not sure about what you would expect there in Australia, but yeah, so that sounds reasonable. Would be interesting to study that in more detail. I kind of regret being here now, although there's like opportunities for looking at what the situation is in Europe, because this species is very much understudied in Europe. It's pretty much been ignored. There are a few papers that have tracked it and they reported on that. But as far as I know, there's not a single study that's focused specifically on Hylurgus ligniperda in Europe. And I mean, it is generally not as common as in the southern hemisphere as you would expect, because there are so many more species in the same niche or related niche in Europe. But then, like in Portugal, for example, we did trap quite a few, also in Greece and in southwest France. So I think there are enough there to do a study and to look specifically at composition. So in fact, if anyone knows of a PhD student who would like to investigate that, I'd be up for that.
Josephine, may I add something to this conversation based on your comment?
Yes,
I saw from Andrew Johnson, you said Ipsgranicolis is interesting. It's a wimpy beetle in Florida, but fills the niche of a facultative pest in Australia. I'm very interested in this insect. One observation I made with my Midwest study, if you remember that graph that I showed from that transect, it was easy to catch its pine in those numbers because we used the pheromone. But the first round we did with just the host volatile cues, and when I actually laid out individual white pine logs, we found very, very, very little Ips pini. It was really only when we had the pheromone that we attracted them. But Ipsgranicolis was very happy and willing to go to those traps with just alpha pinene and ethanol. And if we just placed a white pine log, in the middle of a cornfield in the Midwest, they were almost always colonized by Ipsgranicolis. So, while they're congeneric, there does seem to be some behavioral and biological differences, and it makes me wonder why Ipsgranicolis has been a fairly successful insect, in China and Australia, but Ips pini has never really established across the world. That's something that really puzzles me, and I would love to see more investigation of.
Thank you. We have another question for all the speakers. Any test of algae ligniperda attraction examine chirality of alpha pinene?
That must be Ken Hobson.
Yeah, so we've mostly used racemic alpha pinene and also various sort of degrees of purity, from fairly pure alpha pinene to basically extracts from pine resin. So I mean, the level of attraction to alpha pinene ethanol is phenomenal. So this is as good as you have for a bark beetle like Ips supergraphis to its pheromone, and so that's quite incredible. I think there's still some scope for increasing that. I noticed in some unpublished work the results indicate. that certain bark beetle pheromones increase that further. But we've actually never tested plus or minus, but I believe in pines, if I remember correctly, it's some time ago, but I think it is actually pretty racemic as far as I know.
I do have to thank Ken. He was the one who encouraged me to take the PhD in New Zealand. So thanks very much, Ken.
It worked out all right.
Yes, sir. It was fabulous.
Thank you. The next question is for Lea. How do your results compare with what population genetics patterns reveal of invasion of other bark beetles and other forest insects? Are these patterns unique in terms of numbers and routes of introductions?
So in my thesis, I really concentrated and focused on Hylurgus ligniperda. So I'm not sure about other bark beetles, but I assume there are similar patterns, since other insects can be transported in the same wood packing material as Hylurgus itself.
So maybe I can answer that as well. Thanks, Lea. I'm glad you did get a question, because I was wondering whether it's going to be a bit one-sided, and your very interesting study deserves some questions also. So I haven't thoroughly looked at all bark beetles, but I'm in the process of reviewing this for forest insects mainly. And there is some evidence for a number of others that such bridgehead effects do occur, especially in the case of adjacent or nearby countries where this sort of onward movement to adjacent regions seems to be fairly common. A longer distance dispersal from a bridgehead population to another distant area on another continent also has also been observed. So this would be the example that Lea observed of New Zealand quite possibly being the source of the invasion in California. And there is like similar evidence for a few cases of Syrinx noctilio having been moved around like that. And yeah, but it's not that often been studied in detail with molecular genetic methods. We do know from interception data that there's a lot more potential for that. Like in New Zealand, we used to intercept Ips grandicollis only from the US. And when it became established in Australia, there was an increase in the numbers of Ips grandicollis arriving in New Zealand from Australia on trade leaving Australia. And there's like similar cases that we've seen there. But there haven't actually been that many studies on bark beetles specifically that looked at that with molecular methods.
Thank you. The next question is for Nicolas. Is prescribed fire ever used after clear-cut implantations? It may be too difficult to do logistically, but seems like an ideal way to reduce the quality of phloem resources for beetles.
Thanks, Josephine. Yeah, I agree. Can you just repeat maybe the question to be sure
Yes.
I haven't missed anything?
Yes. Is prescribed fire ever used after clear-cut implantations? It might be too difficult to do logistically, but seems like an ideal way to reduce the quality of phloem resources for beetles.
Oh, prescribed fire, sorry. No, yeah, I wouldn't think that would be much acceptable solution for a species like Hylurgus, which is probably in a sense just a market access concern. The main issue remains with these infestations of logs, and as these logs transit to different locations in the forest, they might be infested by Hylurgus. You would need to perform this prescribed fire at a very massive scale if that would be to be considered as a best risk reduction option. So I think in this context, it's probably way much of a challenge.
Maybe I can add to that these plantations are very large often, and they were planted consecutively over a large area. So you essentially 27 years later or so, one stand is harvested, and then the next one that's right next to it, and then the next one. And so if you had prescribed fire in these places, and you have to remember New Zealand is in the roaring 40s, where it's often very windy, it would be very difficult in some cases to contain these fires. So I imagine it would not go down too well. And yeah, one way that would be good to reduce these populations is to reduce the opportunity for beetles to basically have a huge amount of slash and breed up there. And then the next year, there's another stand fell that's right next to it. And so all the offspring just moves over a few hundred meters into the next stand and has a lot more material. And so that causes this buildup of populations. It would be much better if they harvested one here, and then the next one, say 10 kilometers away or so that you don't have this opportunity for beetles to disperse from one place to another and to become more and more abundant. But the present structure and age distribution of these stands kind of contributes to that.
It would be more around playing around the timing and the distribution of harvest and thinking about where you store these logs to be sure, or at least minimize they don't get infested. Plus other options when that is necessary, like still debarking or some phytosanitary treatments could be applied.
Thank you. We have another question for Lea. So Bernard says, I'm wondering about other examples of connections between New Zealand and the USA, for example, as bridgeheads for forest insects.
I'm not sure if I understand the question correctly.
Do you want me to read it again, or maybe Bernard can rephrase and ask it again?
I think Bernard means if there are other examples where insects from New Zealand have become the source of a population in North America or in the US. And for insects, I would say no, and the traffic has been very much in the opposite direction. There are some plants from New Zealand, and there's the famous New Zealand mud snail that's been pretty good. But in most cases, it's been the other way around. So in New Zealand, there's quite a number of North American insects. Yeah, so I believe there is some potential for more of that, especially of the few insects that are not native in New Zealand. They're very common and associated with something that is exported a lot. But to my knowledge, there are no other cases like that. There's concern about Syrinx noctilio, and it is relatively rare now in New Zealand. So, but I can't remember off the top of my head, Bernard, where the Syrinx noctilio came from that invaded North America. But the fact that there was in the Northeast suggests that maybe Europe is a more likely source. But you probably know that better than I do.
Agree. I think that's more likely.
Thank you. Next question is more of a general question about Hylurgus. So we have two related questions. One is there any biocontrol for Hylurgus? And the other question is, are control measures applied against Hylurgus in New Zealand, or is this not necessarily for economic point of view?
Nicolas, do you want to answer that, or shall I?
For biocontrol, it has never been done, but we found some parasitoids. When was it? 5 or 6 years ago, maybe
Ecki, you remember, or Kevin, you remember this a little bit better.
These were of a species, some parasitoid. I can't remember the species off the top of my head now, but this was in fact released as a biocontrol agent some 40, 50 years ago or so. And then it was never found afterwards, but I think they also didn't look very hard. And then a few years ago, it was actually recovered and found in a forest near Christchurch. And so it looks like this was in fact an establishment after all. And people hadn't looked carefully, studied what actually happens under the bark, and not done any sort of careful observation. But these parasitoids seem to be relatively rare. And I believe it could be wrong that in Europe, parasitoids also don't play as important a role as, say, checkered beetles as natural enemies. Obviously, there is a bunch of birds that are feeding on these beetles, and there are a number of passerines that are very common or abundant even in these pine plantations. But I think they're overwhelmed by the sheer abundance of these beetles, so I don't believe that they actually have any real impact on the populations. And releasing checkered beetles is probably not an option because they're not host specific enough to be considered as a biocontrol agent for introduction.
Okay. I think unfortunately we're running out of time. So Jeremy, would you like to close the meeting?
Yeah, I think it was a great series of talks. I'd like to thank Ecki and all the speakers and everyone who's participated in a very active discussion period. The last webinar is April 1, and this is coordinated by Sigrid Nether on the chemical ecology of Ips typographus and Norway spruce interactions. I look forward to seeing you all there. Thank you.
Well, thanks very much for organizing, and I enjoyed the questions, and I look forward to seeing many of you at a proper conference in the hopefully not too distant future for more chats about this.
All right.
Thanks, everyone. Take care.
Thank you.
Bye, and thanks, Lea, Nicolas, and Kevin again.
Appreciate it. Thanks for the invitation.
Bye. Appreciate it.
Thanks, everybody. Goodbye.
Bye.